So thank you, Amber, for letting me talk a little bit about the work that we're doing at Combined Brain, and specifically the projects that we're working with SLIC 6A1 on. And so today I'll talk really quickly, try not to actually talk really quickly, but about some of the pieces that we're helping SLIC 6A1 and the entire research community on clinical trial readiness. And I'll specifically talk about the importance of identifying biomarkers, whichever those might be, and then the importance of outcome measure selection, which several people have already talked about. Okay, so a little background on what Combined Brain is. So most of you, I see a lot of familiar faces, so this might be old hat for quite a few of you, but Combined Brain is we're a nonprofit. It's a consortium that's made up of more than, I think now we're at more than 52 different patient advocacy groups, and really the mission, the overall mission of Combined Brain is to increase collaboration from the clinicians, researchers, pharmaceutical firms, and researchers, meaning basic and clinical researchers, that are really developing treatments for the disorders that they are researching in their community. SLIC 6A1 in this community here is really an excellent example of that, because some of the research that you're showing today from the pilot clinical trials in Ravicti is really a classical example of when you get a beautiful collaboration between the basic researchers who are identifying compounds, in this case repurposed drugs, and putting them and testing them in a pilot clinical trial and having really extraordinary, awesome results. Okay, so SLIC 6A1 is one of the members of Combined Brain, as you can see here. And really what we've been doing with Amber and the rest of the SLIC 6A1 community is trying to help them move the needle to the right-hand side of this figure, which is, getting testable compounds into kids who need it most. But really, as parents and researchers have provided several examples of this is quite a journey, and is an incredible timeline that goes from identifying patients, getting your diagnosis, to characterizing what the disorder looks like, finding repurposed compounds or novel compounds, establishing a really broad natural history, identifying biomarkers that could be used as outcome measures, objectable measures in clinical trials to determine efficacy. And then ultimately on the right-hand side, let's see if I can have a pointer. On the right-hand side of this figure is what we're all pushing for and definitely prepared for is preparing for clinical trials and having the appropriately identified biomarkers, preferably blood-based biomarkers, or physiological assessments that can be used as objective measures of efficacy again. So what I'll really probably harp on is this bit on the left-hand side, which is the importance of developing and getting patient samples, getting access to these patient samples for basic and clinical researchers to optimize treatments, and to look for these biomarkers. Part of this is developing a really robust biorepository, so an availability of patient-derived samples or patient samples, and this includes a variety of biofluids as well as cell models, and then an EEG repository potentially. So as I was saying, one of the really important things for biomarker discovery is having access to patient samples. And you can imagine in a rare disease community, these patient samples are difficult to get a hold of. But as I saw at the beginning of the day, there were many parents who came up here. And so I will talk to you directly in the next couple slides. But you guys are really, as caregivers, can help participate in the development of these biorepositories that are available right now. So when I mean biorepository, I'm speaking intentionally pretty broadly because, we have a group right now through Simons that is able to collect some of these samples, but really for a rare disease of this nature, you want to be flexible in the sample types that you can collect and distribute to researchers, whichever their research model is that they're using. So this can be cell models, So this I'll say specifically, Combined Brain has actually launched a biorepository, so we're able to collect these types of flexible sample types. And SLC6A1 Connect and has participated in this on a variety of occasions. The table I put together here outlines some of the samples that we're storing for them currently. But the types of samples that we can collect and can make are, we can isolate peripheral blood mononuclear cells, we can generate fibroblasts, we have a reprogramming program, and we're also able to differentiate stem cells into a variety of different neuronal types. We can collect and store blood and various blood components. We're able to collect cerebrospinal fluid. We can collect salivary collection and buccal swabs for people who are interested in looking at genomic signature, genomic profile. And then we're also able to collect urine. We've recently started a program where we're able to start generating stem cells from urine collections for those particular sample populations or patient populations that are difficult blood draws, or collecting blood or getting a skin biopsy is pretty difficult. And then the other thing we're able to store in our biorepository are various skin biopsies or tissue biopsies, and then postmortem tissue, of course. And so here's a table here for the types of samples that SLC6A1 and Amber and her group have already accumulated in the Combined Brain repository. So we have a handful of plasma samples, cerebrospinal fluid samples. We have genomic samples from a variety of these patients, PBMCs, and then we have stem cell lines created from 4 different individuals. And these are all meant to be resources that are readily available to researchers in the community. And we have already distributed these worldwide to many people who are interested in them. So the importance of blood-based biomarkers, several people have already mentioned this, but one of them is really, plasma and serum are considerably less invasive than lumbar punctures, and hopefully your kids aren't having lumbar punctures very frequently. We've set up a really flexible sample collection strategy, so we can collect blood in tandem with clinical labs that are already being drawn in a clinical setting. We have contracts with mobile phlebotomists, so kids or patients who are unable to travel or have a lot of anxiety towards going to clinical settings, we can have a phlebotomist go to your home. And then we have a contract with Quest Diagnostics. You're able to make an appointment there, and you can get your blood drawn there as well. One other important thing that I wanted to remember to mention is that we are also able to bank control samples as well. So several people have mentioned the importance of getting a placebo control or a control arm for a lot of the research studies that have been going on. So we're also able, and we really advocate for, getting control samples from either siblings or parents or other age-matched, unaffected control samples. And then, of course, longitudinal samples as well, every 6 months or annually as feasible. Okay, so aside from biofluid biomarkers, which have been discussed in a couple different presentations here, is also the possibility of using sort of physiological biomarkers. So a potential for EEG signatures. Dr. Goodspeed showed some really promising data that shows that there might in fact be a common EEG signature for the different types of seizures and epilepsies that are commonly present in kids with SLC6A1. And then in an ideal world, have a centralized place to store all of these EEGs to really validate and come up with a common EEG signature. The other potential thing to look at, which would be another objective measure, would be to look at ERPs, or event-related potentials, and really these are a brief explanation of this is just to measure the brain response to any sort of given sensory, cognitive, or motor stimulus. And this is another fairly common objective way to measure the brain's response, and has been used as a validated biomarker in several other rare neurodevelopmental disorders. And then actually, one of the things that Dr. Goodspeed just mentioned is that using neuroimaging techniques or MR spectroscopy as a way to measure select compounds and potentially changes in those compounds in the brain real time. Okay, so a final plug for participating in the biorepository and really increasing accessibility of these critical biofluid samples for biomarker development to use in the clinical trials that are really in the immediate future. So who can participate in the biorepository through Combined Brain? It's, like I said, anybody who has a 668-1 patient. There's no minimum or maximum age range, as well as unaffected siblings and parents. And again, we've intentionally made it as flexible as possible to collect these biospecimens. In particular, blood has been the most popular in tandem with clinical labs, and then Quest diagnostic appointments, and then mobile phlebotomists. And really, we've tried to make this as easy as possible to participate. I know many of you in the room have participated in academic research studies and clinical trials before, and there's a considerable amount of paperwork or other necessary things that are frustrating, and we've intentionally tried to make this very simple and easy and straightforward for families. So really the first step, if any of you parents or caregivers in the room are interested in participating, is just to email our biorepository manager at Combined Brain, and we set it up pretty seamlessly. Okay, so I think that is the plug I wanted to make for the importance of biomarkers and outcome measures. I want to acknowledge, Dr. Bichelle, who's the executive director, unfortunately wasn't able to make it today. Our deputy director, Kristen Hatcher, who I presume all of you will probably meet at 5:30 and never forget. And then the rest of the team here at Combined Brain. Thank you.